a huge success. But we're a country misguided and led astray by media that aren't honest.
My name is Dr. Tony Fauci. I'm the director of the National Institute of Allergy and Infectious Diseases at NIH.
I believe now that we have the agent in hand, that the amount of effort and energy that's being put into it by biomedical sciences, that within a reasonable period of time we'll have a lot of these answers.
Fauci is no stranger to leading the federal response to public health crisis. His career spans 6 presidents and over 30 years of research. In the 1980s, his career heavily focused on the HIV/AIDS crisis.
I'm working directly on AIDS, both clinically and from a basic science standpoint.
He cared for dying patients, met with patient advocates, and has helped shape initiatives to end HIV in the United States.
But if you look at the history of bioterrorism and other types of attacks against the United States and other nations, it dates back to a variety of things, such as in the early 80s, the Tylenol episode, the
In 2001, he helped the public understand the health dangers of anthrax and bioterrorism.
The SARS situation is a little bit more serious than run-of-the-mill, in fact, substantially more serious than run-of-the-mill influenza.
In 2003, it was the SARS outbreak.
Our patient, Nina Pham, is free of Ebola virus.
And in 2014, Ebola. Fauci has spent his career in laboratories, at patients' bedsides, in congressional hearings,
Things will get worse than they are right now.
and on Sunday morning talk shows.
I'm not being muzzled. Everybody who gets on shows like this, you have a normal clearance. I was cleared here, and here I am talking to you, and I'll give you the whole truth the way I always have.
President George W. Bush awarded Fauci the Presidential Medal of Freedom in 2008. Fauci is the second National Institutes of Health Institute director to receive the highest civilian award in the United States.
The 3 people that we now know that were hospitalized working in the lab, do you think Dr. Fauci, because he did have, a research relationship with that lab, do you think he was aware a year ago of that situation, the 3 people had been sickened in the lab? And because a lot of the dogma and the dogmatic statements made by him about this, I don't know how you could have made those if you knew, A, that it was in Wuhan, B, that they were gain of function research, C, that 3 people got sick and had to be hospitalized in that lab. Would you have been as strident as him about the possibility of it coming from the lab at that time if you knew people had gotten sick?
So these genetic testing companies have become quite popular both in the US and in China, but for very, very different reasons. In the US, most people take these tests because they want to know a little bit about where they come from. They want to know their heritage, their lineage, and whatnot. Here in China, though, people want to know what their DNA says about them so that they can plan for the future. If people are, let's say, prone to heart disease or to Alzheimer's, they may change their lifestyles. I'm doing this because I want to know if I'm at risk of having Alzheimer's, like my father.
CRISPR it's certainly a game changer.
CRISPR is a tool that can alter physical traits, even life-threatening diseases.
We can use gene editing technology to let pigs suffer from human disease.
There's something really, really uncomfortable about this.
So for less than $1,000, you can genetically modify humans or human cells.
This gives us the power to control evolution?
Yeah, with this technology, human can do that.
The potential of this genetic editing tool is an ethical minefield that challenges the idea of life itself.
My concern is human enhancement.
Or designer humans.
Yes.
Let's do it, see what happens. I've genetically modified myself with CRISPR. People say, you're playing God, and I love when people say that. I think it's so true and brilliant.
Let's bring in Republican Congressman Jim Jordan. He is the top Republican on the House Judiciary Committee and author of the upcoming book, Do What You Said You Would Do. Congressman, always great to see you. So as you heard Rich Edson say, China's not going to cooperate, and it appears the administration has very little appetite to put their feet to the fire. What's the remedy?
Well, this is nothing new. I mean, China's been misleading us from the get-go. In fact, I just came from a transcribed interview with Dr. Birx, and she said, yeah, China misled us the whole time. So this is nothing new. And frankly, you know who else misled us was Dr. Fauci. I find it amazing, Trace, that everyone in the country wants to know. I think most people in the world want to know, how did this virus start?
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well.
With the help of CRISPR, the floodgates have opened to all sorts of new experimentation previously unthinkable. China currently leads the way in gene-edited firsts, including CRISPR-edited human embryos in 2015.
Do you have pets at home?
No, my wife, they don't like dogs, but I have many fishes.
Unlike the US government's cautious approach, the Chinese government is all in on CRISPR research, providing most of the funding for dozens of the country's CRISPR projects.
Tango, Hercules. We lock down the muscle gene and do not let it work, so the muscle grows without limitation.
Researchers at this lab used CRISPR to isolate and manipulate the beagle's muscle, or myostatin gene, making these the most muscular beagles in the world.
He's incredibly muscular right here in his forelegs, I guess. But aside from being more muscular, everything else is okay? He's healthy?
Okay, healthy, very healthy.
Ready? Let's go.
Oh, these are cute.
These pigs are immune deficient pigs, so they will suffer from infection disease very easily. But with this model, we can do many experiments to do cancer research and immune research and infectious disease research. So this, we use CRISPR to knock out 2 genes at one time, try to make Parkinson's disease and Alzheimer's disease.
Pigs and humans share similar genomes. We have the same immune systems and digestive systems, and suffer from many of the same diseases.
If we succeed in pigs, then we can transfer this technology into human. From the technology aspect, we should no problem to do that now. But so far, you know, the regulation and the law don't allow us to do that.
Seeing all these pigs being CRISPR'd, it is so wild. But there's still something very uncomfortable about this. I think that what makes Americans nervous about CRISPR and about gene editing
For human, right?
for humans. Yeah. But I also think, you know, one of the concerns is because it is a very religious country, CRISPR allows humans, it puts so much power into our hands. It allows us to shape our world in ways never before imagined. And there are many people in the US who think, well, that's not for us to do. That's for a higher power. That's for God. So the idea that we could be playing the role of God makes a lot of people nervous.
Okay.
But clearly not here, though.
Yeah, not that
as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species.
Greetings. I'm Lori Garrett. I'm deeply honored to be here today with three of my personal heroes, and I'm going to introduce them in deference to age. So Anthony Fauci, who is a household name here in the United States, and I'm coming to you from New York City, may not be quite as well known outside the US, but I would say just about every single American citizen at this point not only knows who Tony Fauci is, but has a decidedly clear opinion. And Tony is a household name because he has run the National Institute of Allergy and Infectious Diseases since 1984. And he has guided presidents, congressional groups, and science through multiple outbreaks: HIV/AIDS, Ebola, West Nile virus, Zika, E. coli in food, you name it. Tony has been in a leadership role, and so much so that his opponents are now calling COVID the Fauci virus. And Jennifer Doudna is the newest member of the Nobel Prize Club. She received the prize in the fall of 2020, right when we were all in, let's see, what was that, surge number 2 or surge number 3 in our pandemic, for work that she did together with Emmanuel Charpentier, who is now at the Max Planck Institute in Berlin. They co-discovered CRISPR-Cas9, the sort of genetic scissors that bacteria have used since forever, but humans only discovered when Jennifer and Emmanuel stumbled upon it. She's at UC Berkeley and also the Innovative Genomics Institute at UC San Francisco.
Dr. Fauci, it is good to see you again.
Same here.
You've been doing amazing work. You've got to get it in, and the weather is improving. I should tell you that I love running outside as long as it's above 50 degrees. So,
you know, you and I are right on. I'm a wimp. Anything lower than 50, sorry.
Not gonna happen. I see folks in shorts running in 40-degree weather. They're insane. I know that you were involved in the various groups that helped to develop this set of vaccines. Tell us a little bit about the process for the creation of the vaccines. How is it that we were able to develop vaccines as quickly and vaccines that are as effective as they are? Because historically, it's been taken a lot longer to develop vaccines for a disease that is this severe. What was it about the process that allowed us to do that? Is it technological? Does it have to do with new methods, new systems, and how was your office involved in that process?
Okay, so that's really a critical question that I believe the general public doesn't fully appreciate. So the virus was identified and sequenced and made known to the public in the first week in January of 2020. And 11 months later, we had vaccine going in people's arms. That is totally unprecedented. It usually, as you say correctly, years before you get a successful vaccine.
In research, it's not that often that when a big real world problem happens that you can actually do something about it. And this is a case where we as scientists, we can actually directly use our skills to do something. I think it was after seeing some of the news coverage that the virus is really spreading, and also seeing that there really is a deficiency in tests. We know what we're looking for in these viruses, but we just need to figure out the right combination of things to put into the reaction.
The U.S. authorization of 2 COVID-19 vaccines marked a turning point in the pandemic and vaccine science. These vaccines use messenger RNA, or mRNA, to deliver immunity. mRNA are short strands of instructions used to build proteins. It's impossible for these mRNA vaccines to alter our DNA in any way. Although that technology has been in development for decades, mRNA vaccines hadn't made it to market, until now. There are a few ways to make a vaccine. Most use a version of the pathogen they are trying to protect you against, like the flu or measles, to trigger an immune response. You can think of vaccines like a biological wanted poster. They show our bodies how to recognize the enemy, an antigen such as a harmful virus or bacteria, so we know how to react if it ever shows up. Our bodies will be prepared to send T cells to attack infected cells, and B cells to produce antibodies, which neutralize the virus and prevent it from invading healthy cells. mRNA vaccines use innovative technology to deliver protection. If traditional vaccines deliver wanted posters, these vaccines carry USB drives with instructions that tell our cellular machinery how to print its own poster. In other words, they instruct the body to make its own antigens. In the case of the coronavirus, the antigen is the infamous spike protein that attaches to and infects your cells. These vaccines use mRNA, a molecule that our cells need to create any type of protein to introduce us to that spike. To start, researchers isolated the part of the coronavirus's genetic sequence that codes for the spike protein. Then, they put an mRNA molecule containing the code into a super tiny drug delivery system known as a lipid nanoparticle, which can enter our cells. Inside the cell, ribosomes use the code to create a protein chain that resembles the enemy, the coronavirus's spike protein. Those cells can then temporarily display a lookalike of that protein, which prompts our immune cells to respond as if it were an antigen from a traditional vaccine.
Early on, I thought, I'm going to write a book about the great adventure of understanding gene editing. You know, I've written about the physics revolution that dominated the first half of the 20th century. And then, of course, I was deeply immersed in the digital revolution, which, was the second half of the 20th century. But what happened in the past few years is we found easy to reprogram tools that will allow us to edit our genes. Man, that's going to be 10 times more impactful than the digital revolution was.
We all want to return to the way things were before COVID-19. But there's one area where I hope we never go back: our complacency about pandemics. We can get ahead of infectious disease outbreaks. By the next pandemic, I believe we can have what I call mega testing diagnostic platforms. They can be deployed quickly, cost very little, and test 20% of the entire population every week. We also want to get treatments out far faster next time. One of the most promising is monoclonal antibodies. These manufactured antibodies grab onto the virus and disable it, just like your immune system, and can reduce death rates by as much as 80%. I also think that we'll develop new vaccines quickly, in large part due to this new mRNA platform. mRNA will become faster to develop, easier to store, and lower cost. That's a huge breakthrough. To stop future pandemics quickly, we need to be able to spot disease outbreaks as soon as they happen anywhere in the world. And that requires a global alert system. If there turns out to be some new infectious pathogen, then we need a group of infectious disease responders to spring into action. Think of these as like pandemic firefighters. They're going to use their logistics, use their ability to build up capacity quickly. They're going to go wherever that problem is. Stopping the next pandemic will require a big investment, But I think of this as the best insurance policy the world could buy. You can read more about this in our annual letter.